Okay, I'll give you a talk on the causality. These are my main collaborators I'm working with, mostly Anas, Fernando, Aaron, and also a lot of thanks for the NIH and the NSF, actually. So the data we're going to analyze is the resting state MRI. And we use Pearson correlation. We actually use a time-lapse Pearson correlation detected with directionality. And we use about 20-second window. Now, I'm actually showing you about 100 image slices of the animation, but the projector is not able to display it properly, so it's very slow. Actually, it's each slide supposed to be compressed into. And anyway, it's just showing that this is what the usual resonance state fMRI dynamics looks like. It's essentially white noise. I would say it's about 95% white noise, about 5% signal possibly. So if you look at this fluctuation over time, it's the connectivity. If you try to align it temporarily, that's nonsense. That's always nonsense. You cannot align it. So because it's nonsense, I'm not doing it. Okay? I'm going to show you how to align this temporarily. Okay? That's the key message in this talk. It is possible. I believe it is possible. I haven't published it. It's all primary research we have been doing for almost like 1 year. We are in publication. How? Through topology. Even though there's a fluctuation, it looks like almost white noise. I believe there's underlying topological backbone that's stable, and we're going to average this and show a new result. So what's the problem with the traditional causal model? There are a lot of causal models, gradual causality, structure equation model, and you might be familiar with this dynamic causal model, which is Carl-Christensen's work. Now, all these models are based on equations. Okay? So the region J will be here, will be equated as a linear combination of all other given in the past time form. Yeah? What's wrong with this very nice equation? It's very nice equation. It works. The problem is that this is equation you cannot represent or identify cycles. Yeah? Almost all the causal models are built on top of pre-data structure. You cannot get this kind, you cannot model this cyclic structure. And this is the biggest problem with all these equations. So, I mean, even though this is a nice equation, you can extract the signal. All the signal you want to extract out of this equation is most likely free structure. You cannot get the cyclic information. So there is no such thing, no such concept of the cyclic causality. You introduce as a mathematical nonsense there. Yeah? Another causal model would be popular model with Power's model. Yeah? Power's model basically use a conditional property. Instead of setting up the equation, you put a conditional property, yeah? Condition on the past event, you put a problem. What's wrong with the conditional property? Again, same problem. You cannot put conditional property on top of conditional property in a cyclic manner. It introduces mathematical nonsense. Right? So all these causal model cannot be formulated on the cyclic graph. That's the biggest limitation of all the causal model. So if you're using this model to analyze brain network dynamics, what you want to get is it's like a black and white picture. You're essentially missing the big component of the network dynamics, which is cyclic. So we need a better method. So one method can handle this kind of cyclic structure. Obviously, topology. we're gonna do a little bit of the topology data analysis, which is what we call partial homology. This is my paper when I written when I was an assistant professor a long time ago. This was actually first partial homology paper in a brain imaging. And two Peters, I called him two Peters, taught me this topology. And for last 14 years, I've been working on this problem for many. And I think I published like 50 papers on the TDA topology. Now we got this one. If you know something about the Gaussian homology or the whole data analysis, most people use this kind of diagram. It's called Gaussian diagram, which is a kind of summary of the topological declaration. This is very useless in the neuroscience or neuroimaging. Only give you the pictures, summary of what's going on. It's not a very good tool for quantifying causality, but we can make it to work. So we're going to start with some sort of the structure or algebra. So we're going to build use simpler complexes to represent the network dynamics. In this case, what you're saying is the time-lapse case correlation. We use the time-lapse correlation to identify the directionality of how the one brain region will be affected by another. Now, this one is pairwise interaction between two nodes. What about the interaction between three nodes? You need three edges. So instead of doing that, we want to represent the interaction of the three nodes as an object, geometric object, where this will be encoded as a triangle. Okay. Four node of interaction will be simply encoded as a tetrahedron, as a single object. Now, this kind of conceptual simplification makes things a lot cleaner and eventually get job done. So this is how we're going to do. And for this one, we need some mathematics. And the basic method we explained in this paper is transactions and the medical imaging. And about 2 years ago, he's my former postdoc. And so you do this kind of operation. And the one thing I want to point out is what the boundary is. It's basically nothing more than incidence matrix in graph theory. It's a general distribution incidence matrix explaining how the low dimensional structure is combined to form a high dimensional structure. In this case, how all these edges are combining to form a sigma, which is a triangle. So these three edges will form a triangle, and you can put it. So you can put a directionality of plus and minus the directionality in the simpler complexes like this. Then with this, we can build what we call the Laplacian, and it's called the forced Laplacian or the combinatorial Laplacian. It's an amazingly powerful tool. If you're working on the network or graph in general, I strongly suggest you to embrace this new Laplacian. This is a much powerful framework than the usual graph Laplacian. The graph Laplacian is defined on it's like a relation between node to node. Okay? While Force Laplacian can establish a relationship between high order structures, between edge to edge. So the first Force Laplacian is defined along the edges, while graph Laplacian is defined on the node. So that's the difference. And you can actually generalize this into high dimensional Laplacian, Gauss Laplacian as well. And with that, so it's just how the Laplacian is constructed with the incidence matrix or boundary matrix. So B1 is basically the relation between the node and how it's formed to have edges. B2 is basically how the edges are connecting to form a basis. And then, I know this may be a bit tedious, but we need it. Now I'm going to build the energy. This is one possible way. I thought this is most clean way to do it. We can think about a slightly different energy term, but I'm going to stay with this. Direct energy usually it's defined on the node level with graph Laplacian. We're going to extend it to the Hose Laplacian and we're going to define on the edges. So with that Laplacian, Hose Laplacian, and X is basically the edge flow. So once you have the connectivity, you're going to factorize it and only get the edges. So that's factorized version of the connectivity. And you can form this kind of direct energy. The first term measures the divergence, which is source sensing. Now, this is what the graphical Laplacian can also measure. But what the graphical Laplacian cannot measure is this cycle. It's a third term. It's not a force, it's a source sensing. Sorry. It's a typo. Please ignore this. The scar is basically circulation around the three node. So that's what we have. If you want to incorporate some sort of anatomy, we can also have this kind of, you can modify this Laplacian. This is just a concept. I haven't done anything. It's just an idea. I'm working on it. And where the C is, you can think about this conductance. And you can come from like a 5 count, for example. This is the example of the fMRI connectivity superimposed on top of the 1 meter fiber. So we can do something like this and construct the Dirichlet energy from this 1 meter fiber and combine this together. Multimodal fusion way. So the energy term can have the anatomic term and functional term together. So you don't have to do anything. Yeah. Then this is the core of this talk. So basically, energy will form. Energy will basically flow from highest energy to lowest energy. That's the idea. That's how I do the model, the causality. The simplest possible idea. Okay? And you just follow this, just do the derivation and follow the steep gradient and just solve it over time. And that's it. So if you solve this partial differential equation, that's the causality. It's a Dirichlet diffusion equation, basically. And what will happen is that if you have this kind of edge flow, if you run this to the infinity, that's the exponential term will that's basically come for the diffusion, basically. And the pH is a projection operator for the harmonic. You basically projected the edge flow to the harmonic subspace, basically. And so you have the edge flow will move all the way to the harmonic. That's the steady state, the harmonic flow, or the steady state. And all our terms are disappearing. It will disappear if we run the diffusion enough time. And you can solve this in, you don't have to solve the partial differential equation. You can solve this by simply doing a matrix multiplication. Yeah? The nice thing about the harmonic flow is that my causal pathway is essentially harmonic flow of the signal, fMRI signal, is that whatever perturbation you put in the harmonic flow, it will stay in the harmonic flow. Harmonic flow is basically residing on what we call the harmonic subspace, which is vector space. It's very stable. Whatever you do it, it's again in the harmonic space. Okay? So that's where the power of the stability comes in. Whatever perturbation you put it in a harmonic flow, it will be harmonic flow. Okay? And since it's a vector space, I can do all those step operations, summation, and variance computation, etc. And performance is just remarkable. Whatever you put it, I can challenge you. Whatever you put it, whatever you bring to me, whatever program model you bring, I can do it. As long as the network has cycle or the cycle. Okay, usually performance is 10% gain over existing methodology. It's no argument, no competition at all. And we apply this our form subject and point window and to see how the harmonic flow changes over time, and it's about 40%. So and additional term is about the 5%. So about 45% of the rest of the MRI is essentially affected by the cyclic infection. And why people never discovered it? Because they never applied pathology. It's that simple. There are tools, all the tools people analyze is based on tree structure. So they never look at it. So they never want to find it. If you assume you use the topology, you will able to find the cyclic infection. Change your tool. That's what I always suggest to people. So this is the result. So if you average the time correlation over time, you get essentially randomness. You're not going to get any signal, but this is the average over time, and also average of the form structure. I guess it's so stable. This is what I call the topological backbone. Now you can see it's a symmetric, left and right symmetry that cannot possibly happen in the randomness. Why it's a symmetry? It's a cycle. What you're seeing the collection cycles. Why? It's a motor sensor. Motor area. It's a left-right hemisphere symmetry. Our visual area symmetry. So all this should be cyclic. I don't think it. So that's what you're getting. And so I thought this cannot possibly random or fluke. So I actually worked on so for last couple of two days I was working on whether there's actually mathematical justification. Actually there is. Actually I found a new theorem. So it's a spectral variability of the harmonic flow is far smaller than the variability of the actual edge flow original data. That's why I have to actually detect signal. And that's it. I'm done with this talk, and thank you. Yeah.
Any questions?
Yes. Yeah.
So wondering what about time? Meaning you're assuming all arcs have.
It depends on time because the cycle. So I have to compare in the 20-second with sliding. Within 20-second sliding window, we have a cycle. That's So if you change this scale to like 0.1 second, I don't think you're going to capture anything. Everything, I mean, at the neural level, everything is just a tree structure, not much cyclic. However, if you increase the scale, you're going to observe all this interaction will be forming cycle. So it's just the scale dependent. Yeah.
Intuitively, I think it makes sense that the brain keep being stable in resting state. I wonder if you repeat the process in like a class-based MRI or in a disease state like epilepsy, what would you expect?
Oh, actually, I did some modeling. It's a counterfactual modeling based on actual. It's a healthy population. Based on literature, I've identified some, and I actually with the actual healthy population, I actually thought to make it disease basically, and we can do it. Now, if you get this is harmonic flow, healthy flow. This is a model. My model of this is my model of temporal epilepsy. If you look at it, I can actually change it. If you apply some sort of operation, it's temporal lobotomy. You cannot do it in a healthy subject. You cannot do it. I just do it in a computer, and it doesn't change because it's harmonic flow. So it's very stable. Even in the disease population, there will be disease specific harmonic flow. Yeah, but we can talk about this later.